Okay, finally in this third video, we're going to talk about the action potential, how the muscle cell is actually excited. So this is one of the properties that muscle cells, or otherwise known as muscle fibers, have, is they have this ability to be excited. And the reason they have this ability is because the cell is polarized. So a lot of the things that I'm about to say are going to sound just like what I described for the neuron. And I think that's okay. They're cells, they're similar, and the fact that you've learned this about the neurons is going to make it much easier to understand about the muscles. So first thing we need to understand is that a muscle cell at rest is negative inside, meaning there's more negative molecules inside the cell than outside. So this causes polarity, kind of this negative positive difference across the cell, just like a battery will have a negative and positive diode, and we say a battery has polarity, so we say for a cell. The muscle cell has polarity because it's more negative inside and more positive outside. At rest, its membrane potential is down here at -70. And the reason it is negative, there's going to be several things. One, there's going to, because of the selective permeability of the membrane, large and charged molecules can't move across. And inside the cell, there's going to be lots of slightly negative proteins. All those myofibrils are slightly negative. So that gives the inside of the cell a large negativity because we have negatively charged proteins that are big and trapped inside the cell. The second thing that's going to affect the membrane potential is a sodium-potassium pump, which is this protein that sits in the membrane and does this job of pumping sodium out and pumping potassium in. So this creates these concentration gradients of sodium-potassium and creates a story where inside the cell we have a lot of potassium and outside of the cell we have a lot of sodium. And yes, it's exactly the same as what we saw for neurons. And then to make it even more similar, the other thing we have is once we pump sodium and potassium to their relative positions, so we have a lot of sodium out and a lot of potassium in, the cell also has potassium leak channels. So these are channels for potassium that are always open. That's what we mean by leak. They're not gated. They're just always open. So this potassium that the sodium-potassium pumps in can go right back out. And that, instead of causing the cell to be a little bit positive because of the potassium being slightly positive, causes the cell to be even more negative as that potassium also leaves. And it actually usually sits right outside the membrane, causing this big difference in polarity directly across the membrane. So, a lot of the story, like I said, is just like the neurons, but it creates a situation where the muscle cell is polarized. More negative inside, more positive outside. And that will be at rest. So at rest, the cell is slightly negative. It's going to be this -70, and that would just kind of chug along there at -70. But then what happens is when you stimulate a muscle cell, it's going to cause these so stimulation of a neuron causes gated sodium channels to open. So what we will eventually talk about is in the membrane of muscle cells are these channels. They're called ligand-gated sodium channels. So we're going to have these ligand-gated sodium channels, which should definitely sound familiar. Sodium channels. And the ligand will be acetylcholine. We'll eventually get here when we talk about the neuromuscular junction. But what's going to happen is acetylcholine is going to be released near the muscle cell. It's going to bind to this ligand-gated sodium channel and cause the sodium channel to open. When it does, here's a better picture of it opening, sodium will rush in, bring with it the positive charge, and cause the cell, instead of being -70, to be something more positive as all the sodium molecules rush in. This is called depolarization because we're losing that polarity across the membrane because inside the cell is becoming more positive. And so now there's no longer a polarity. This is depolarized as sodium rushes into the cell. Then the next thing that's going to happen is these sodium channels will close again. The acetylcholine will be cleaned up and these sodium channels will be allowed to close. And when it does, the sodium-potassium pump is still working. It actually never stopped. So it's effectively going to pump that sodium back out. And while doing so, it'll pump the potassium in, but that potassium just goes right on back out. And this allows the cell to go back to being negative. This is called repolarization. We get this tiny blip of hyperpolarization because of more potassium leaving, because we're actually going to open a few voltage-gated potassium channels to help get the potassium out really quick. But this causes the cell then to repolarize and go back to rest. So we saw that it's going to be acetylcholine that binds to these ligand-gated sodium channels that allows the sodium to go in. Let's see where this acetylcholine is coming from. The answer is it's going to be coming from neurons. So there are going to be neurons all along the length of the muscle fibers. And these neurons, specifically, I'm talking about the axons of them. If we look again at this picture of the muscle, so here we're seeing a whole muscle. So this is going to be epimysium, which is going to surround these muscular, these bundles, which are the fascicles, which are going to be surrounded by perimysium. Inside these fascicles will be the individual muscle fibers surrounded by endomysium. So we've kind of seen all this before. And if we watch, though, what the new information here is going along the outside of the whole muscle are going to be these axons, which are part of a nerve. So these axons will branch, or the nerve, the axons inside will branch, and some of the individual axons will come out and continue to go along the muscle fascicle. And then you'll get some of these axons will go all the way down to the level of a single muscle fiber. And so here we're going to follow these axons as they're actually reaching the individual muscle fibers, which are inside these bundles or these fascicles. So here, these are the individual muscle cells, and here we're going to be seeing the muscle fiber hitting it. So the axon branches within the perimysium, such that one motor neuron, so these are motor neurons, will synapse on more than one muscle cell. And anytime you have a motor neuron synapsing on a muscle cell, it's called a neuromuscular junction. And we use this word junction. We could have used the word synapse, which is where we've used everywhere else, but at the muscle, for muscles, it's called the neuromuscular junction instead of the neuromuscular synapse. And we're going to describe at this location where the motor axon, the terminal of the motor axon meets the muscle, is called a motor end plate on the muscle cell. So let's see if we can look at that a little bit closer. In this picture, we've way zoomed in. This is the neuron. This is its axon terminal. And at the axon terminal, you're seeing these vesicles full of the neurotransmitter, which is acetylcholine. And then we're seeing this is the muscle fiber. So this is its cell membrane. And you can see we've got some, they're showing the charge difference, which we'll follow in a minute. But this is the muscle cell membrane. And then here at the neuromuscular junction, the motor end plate, what we mean by motor end plate is this area where the muscle fibers sarcolemma is highly folded. So at the neuromuscular junction, here's the neuron terminal, and here's the muscle fibers motor end plate. So the motor end plate is just this highly folded area of the muscle sarcolemma. And it's folded like that, so there's lots of spaces for the acetylcholine channels, so that we can have lots of acetylcholine interacting with these acetylcholine channels, opening them. The acetylcholine channels are ligand-gated sodium channels, so when acetylcholine binds, they're going to open and sodium will rush in, depolarizing the cell. So the neuron releases acetylcholine. Acetylcholine binds ligand-gated sodium channels on the muscle fiber, specifically the motor end plate of the muscle fiber, where there's a big concentration of these ligand-gated sodium channels. When it does so, sodium rushes in, and that's going to bring in this positive charge instead of a negative, to get rid of the negative charge. So this is called depolarization because the sodium's bringing in these positive charges. If this changes the membrane potential to a threshold, at this location, at the motor end plate, if we reach a threshold, we're going to open additional voltage-gated sodium channels that are further away from the motor end plate. So we've just got to change the membrane voltage enough here at the motor end plate to then open the voltage gated channels. And then all along the rest of the the muscle cells membrane, the sarcolemma, are going to be lots of these voltage gated sodium channels. The idea being, just like it was we propagate an action potential along a neuron cell membrane, in a muscle cell membrane we're going to propagate it in the same way. The story will go, if we open a voltage gated sodium channel, sodium rushes in, we get a change of voltage here, that'll open the next voltage gated channel. Sodium will rush in, we'll change the voltage here, that'll open the next voltage gated channel. And this will continue all along the length of the sarcolemma, where we have this depolarization. And then in its wake, we'll have the repolarization as we kind of pump that sodium back out and allow the cell to repolarize. Here in this next picture, this is showing that propagation that I tried to draw previously. This is showing that we're going to open sodium channel, sodium rushes in, it becomes positive here. But then then that will open the sodium channels in the next place, sodium will rush in, and that'll open sodium channels in the next place. So we'll watch that action potential move along the length of the fiber. That's the first thing you can follow. The second thing that you can follow is in its wake, we're going to have the sodium-potassium channel pumping sodium back out, and we're also going to have voltage-gated potassium channels open briefly to get more of that positive charge back out. This is going to be part of repolarizing. We open the potassium channels because that helps. Remember, there were already potassium leak channels. So potassium was already leaking out, but now we open additional potassium channels. That gets even more of the positivity out. The sodium is also being pumped out, so that's trying to get that positivity back out and get the cell back to this resting membrane. Eventually we close back up those additional potassium channels and we have restored the resting membrane potential. And so that's going to happen consecutively in each area of the cell. So we have this wave of depolarization followed by the wave of repolarization. Now remember, for a muscle fiber, this action potential will be able to travel along the sarcolemma, but also into these T-tubules, which are these invaginations. And so with it, it's bringing this change in voltage. The T-tubules are adjacent to the sarcoplasmic reticulum membrane. So in this picture, we're seeing, I'm going to try to outline, this is an area of sarcoplasmic reticulum membrane here. And sequestered inside it is going to be all of the cell's calcium. We're also seeing sarcoplasmic reticulum here, and they're not illustrating it, but it would also be over here. So we always have sarcoplasmic reticulum adjacent to the T-tubule, because in the membrane of the sarcoplasmic reticulum, there are going to be voltage-gated calcium channels. And so what's going to happen as the change in voltage is spreading up the T-tubules is this change in voltage is going to be right next to the sarcoplasmic reticulum membrane, which is where you have the voltage-gated calcium channels. So this is the membrane potential then in the T-tubules causes these voltage-gated calcium channels to open. And when they open, calcium spills out into the cytoplasm, which will then interact with these. So then the calcium comes out into this area. Calcium is now just free within the cell or within the sarcoplasm of the cell. And that's where the myosin and actin are. And then the story goes, the calcium binds to troponin, and the troponin moves the tropomyosin so that the myosin heads can now bind the actin and do that. And then the myosin heads in the presence of ATP will bend and pull the actin toward the end line or the middle of the sarcomere. This is kind of a big deal, so I'm going to repeat it a couple times. And just always when I repeat myself, I worry that it makes it sound like it's more information, when really I'm just trying to say the same thing, maybe with a little bit more clarity or so that you can see it multiple times. So in this picture, what we're seeing is this is the voltage traveling up the voltage change traveling up the sarcolemma, opening consecutive voltage gated channels, allowing sodium to rush in, changing the voltage, opening the next one, allowing sodium to rush in, changing the voltage, opening the next one. It travels along the sarcolemma and down the T tubules. This causes a change in voltage inside the T tubules, or across the membrane of the T tubules. This change in voltage is felt in the sarcoplasmic reticulum, which in this picture we're seeing kind of as all of these blue cisternae. And trapped inside the sarcoplasmic reticulum, we can kind of sneak inside and see there, are all of these little red dots, which are the calcium molecules, or the calcium ions. And so when we have this voltage change here, it causes these channels in the sarcoplasmic reticulum membrane to open, and calcium then spills out into the main part of the cell. So now we're spilling calcium out into the main part, and out here in the main part, this is where the myosin and actin are. So as soon as these little calcium molecules are released, they can go and interact with the troponin and the tropomyosin on the actin filament, so that the myosin heads can bind. Now, one additional thing that I'm able to describe in this picture, I like this picture because you can see it fairly well, and I'll show you again in one more picture after this, is a conformation called the triad. And what the triad is around the T tubules, you're going to have two adjacent sarcoplasmic reticulum. They're called the terminal cisternae of the sarcoplasmic reticulum, but they're kind of these wider areas of the sarcoplasmic reticulum. And this is called a triad because we kind of have the three things in the row. We have the T tubule, and then we have these wide parts of the sarcoplasmic reticulum, and they're all located right together. So this is called the triad. And we're going to see that again in this next picture, where we're kind of illustrating the same thing, but we've zoomed out a little bit, so maybe you can see this picture, more of a big picture. So to kind of orient you, we're looking at a muscle cell. So this is a muscle cell. It's called a muscle fiber, and it's packed with these myofibrils, which are all of these myofilaments of actin and myosin. And so if we kind of zoom in here, here's bundles of myofibrils, which are bundles of the myofilaments. And so the little filaments are these little dots inside of here, but they're the actin and myosin. And maybe here we can kind of see, here's the myosin attached to the M line, and here's the Z disc, which has the actin attached to it. And so this is inside the myofibrils. And then around the outside of the myofibrils, wrapping the myofibrils, are the sarcoplasmic reticulum cisternae, or hallways, or rooms, or compartments. So in purple, we're seeing all the sarcoplasmic reticulum. And locked inside of here, I don't really have enough room to draw, but trapped inside of here is the calcium. It's trapped in the sarcoplasmic reticulum membrane. And so what we can see happen is, as an action potential, a voltage change spreads along the sarcolemma. It travels also in both directions, continues along the sarcolemma, and travels up the T tubules, bringing this change in voltage, which is felt at the sarcoplasmic reticulum, change in voltage, which opens the voltage-gated calcium channels in the sarcoplasmic reticulum membrane, spilling calcium out, and now that calcium would be able to interact with the myosin and actin. And so this is called excitation, meaning the voltage change. Excitation, contraction, coupling. And the secret to the coupling is that voltage change opens the voltage-gated calcium channels, which causes the calcium to be spilled out. And then when the calcium is out around the myosin and actin, the calcium interacts with the troponin, which moves the tropomyosin, which allows the myosin to bind the actin and do those repeated contractions as long as ATP is present. And then as long as the calcium is out and ATP is present, the contraction will continue and the muscle will go into this contracted conformation. Then, meanwhile, if there's nothing keeping the voltage-gated calcium channels open, then that calcium can get pumped back in, also with ATP. We pump the calcium back in, and as soon as we get all that calcium cleaned back up, then the troponin will allow the tropomyosin to recover those binding sites, and then there'll be no more interaction of myosin and actin. The cell will go to rest. Whew. So this is how the cell contracts. Oh, and a password. So we need a password this week for the quizzes, and we'll use calcium as a password. Let me check that that has the right number of words or letters. 1, 2, 3, 4, 5, 6, 7. Yes, this should work just fine. So our password this week is calcium.